Yeah.
Okay, all right. So just briefly tell you the history. The whole idea of GIBEX came from a large program which I was fortunate to be a part of, run by National Institute of Health, called ICBT, International Cooperative Biodiversity Groups. And that program was to discovering new pharmaceuticals from conventional products, working with international partners. Basically, what it was what we call bioprospecting, where we're supposed to collaborate with countries after signing all the agreements according to the agreement mentioned, and so forth, and then work with developing countries and with industrial partners to try to discover novel agents. And we were working in Central Asia, we had a program there. But what we quickly realized, and that many of my colleagues did too, that the idea of going to a foreign country and collecting their plants is not something which is favored by local people or governments, because it's often been equated by piracy or recreation. That kind of bioprospecting did not really work very well all around the world, because for a variety of reasons, it was perceived as Western countries, developed countries, trying to take resources. So in response to that, and in collaboration with several universities in the US, University of Illinois, University of North Carolina, University of Rutgers, we have decided basically to change the paradigm of how we discover novel biologically active compounds on next-gen products. And we call it the reversing the flow paradigm. So rather than going and collecting plants and doing basically by assays, what we said is, can we actually develop very simple ways to collect plants in the field, within the country, with local scientists, create extracts in the field, even without the need of the laboratory, and also develop a very simple leak detection pharmacological tests, which can be operated by scientists in any country in the world in the field, which doesn't require sophisticated lab. Can we simplify this whole process and therefore enabling all the discovery to be done in the host countries rather than taking materials out? And this was initially started in Nigeria in 2005. We tested this idea, we started to developing the simple ways of extracting plant material, simple way of testing it, so it can be done in the fields, and the marshes, and the jungles in the country, so the country actually can claim complete ownership of all the results of which happen, and then they can expose it. So that idea was really supported by the Nigerian scientists, and slowly but surely. And that's where GIBEX stands now. The simplified method of collecting and testing field materials under the GIBEX is now in actually 28 countries of the members. And we've done active workshops where we'll go and together we'll teach actually the techniques we developed, and I will describe them later, and the methods we developed and testing we developed, share them with scientists that are usually developing countries. And Indonesia is a very developed country, so it may not be perfect for it, but many African countries and some South American countries, this was a wonderful tool, not only to discover the leads, but also to get students interested in science and giving them the joy of discovery. So 16 countries in red, this is where we actually have active workshops and work with scientists and 48 other members. So obviously GIBEX have evolved, and our way of field testing biological materials and structure have evolved. But as of today, basically there are two components to this. The first one we call RAMES, and this is rapid metabolite extraction and storage. It's how do you, without sophisticated laboratory facilities, you go to the field, basically from the truck of the car, can extract materials, immobilize them, and basically create a metabolomic library, which subsequently can be used for testing in something what we call screen-relation technology, because it's uniquely adapted to work with the format in which you collect plant extracts. So the advantages, and then it was tested in many different countries and perfected, and this is what I'm going to show you today. This is my graduate students and postdocs that travel around the world, work with different communities, really craft, I work with Morani natives in Ecuador. Sarah Skubal teaching Rangers at Kruger National Park, up with actually use the field extraction testing technology on the RAMES. And this is how it looks at the bottom left. This is the trunk of the car. Everything has been run on the trunk of the car using basically rechargeable devices. You don't need a wet lab for anything. Is it a good way to discover good drugs? Probably no, but it's still a way to discover biological activity effectively for many applications and also for education. I mean, we have great experiences teaching high schools in North Dakota and Alaska, in the US, who inherited a lot of traditional knowledge from the ancestors, but they were able to verify. This level that go to the field, look at the plants, like grandfather stolen, mortars, that's and whatnot. You quickly see that we've got a lot of people interested in science. And it's very low impact. So it's basically a way to bring the laboratory to the field and bring this joy of discovery to younger generation of scientists, hoping that this will be followed on and you can really build science based on this initiative. So how do we do this? Okay, so this is the process of RAMES as it stands now. Okay? It only requires 2 grams of plant tissue. And of course, the key thing there, my colleague Dr. Brown will tell you how important it is that before you do that, you really validate the source. I mean, you have to do a variant voucher. Of course, we'll tell you how to do DNA sampling, but knowing what you're collecting, that's absolutely key. Many different ways to do it. This is not a subject to the store, but you need to know what to do. So 2 grams, if you weigh the 2 grams on the balance, which is battery operated balance, then they use those chambers, which are basically the keyhole chamber. You can buy them in any art store supply. And we've adapted, and this is really sort of one of the Dremel tool. It's a bust revolution tool, which you can buy in any university in basically in the world. And the only thing which we added to that is that blade, which is really we developed, which is very effective and polarizing of grinding plant material in 70%. This is what we do. 5 milliliters to 70% ethanol, put it inside the chamber. That blade goes inside, and this is all aluminum. And within 20 seconds of just grinding it, we have, and when we supply the tools, we supply Dremel, we supply a bit, and we supply that lid and chambers at very expensive, couple of dollars each. So many of those now around the world are used. And so it takes about 20 seconds, 15 seconds to grind the material in the field. You get a very nice extract. You get, we prefer 70% ethanol because it's available in many countries. Actually, all of it is published. It was a post one in 2018. I mean, all the pictures of them. I'll be happy to share. No, it's published bilaterally. And then again, everything we use is off the shelf. So we use the seeds, which we buy on Amazon. Funnels, circulation vials. In the field, you can quickly put your extract on basically this plastic mesh filter. You press it with the bottom, and you get your extract collected in the scintillation vial. Again, you have a way of doing it with bark, which is just using different blades. I don't take them. And then I think this is a really important thing: is how do you quickly dry and immobilize and protect your bubble of your biochemistry of the plant. For that, after doing a lot of searching and again, some of that is published, we've discovered that glass fiber filters, okay, which is very inert material, you can buy big sheets and then sponge those circles out of them. What we do is we just spot 100 microliters of an extra on the surface of the glass fiber disc. And that very rapidly captures all this. So you're only like 20 seconds away from extracting the material. You put 100 microliters on the glass fiber disc, then you put them on a little needle and then just drive them in front of the USB rechargeable fan in the field. Okay, so now every disc contains the immobilized, basically metabolome, like 70% ethanol extracted metabolome of your sample plant, and it takes maybe 2 minutes to dry. So rather than collecting big batches of material, extracting it, and creating this gooey sort of extra in the bottom of the glass tubes, now we have the extract of the metabolome mobilized on the surface of the glass fiber discs. And this is how it looks, for example. Okay, it's put in an envelope, you have to label it. And so this is a much faster, much more efficient way to extract plant metabolome, and it's non-destructive because one leaf is actually sufficient, right? 2 grams is not. You can produce about 20 discs impregnated with biochemical compounds from each particular plant, marine organism, which is what it does. So again, it's called RAMES, so it's metabolome and genome technology, because we believe it's very important also. In addition to taking good pictures, again, this is all teach how to document, you can write about it if you need to, but also, and this is something we didn't develop, but which is adaptive. If you punch the leaf disc, okay, and you put it in a plastic bag, a Ziploc bag with silica gel, it will dry very rapidly, sort of a very good DNA voucher. Again, it takes a second, so you have your you punch a disc at the same time as you extract the plant. You put it inside of the Ziploc bag, you put silica gel in it. In 5-10 minutes, it's dry, and you just store it with sand. So now you have the DNA from your plant, so which you can use for identification, validation, whatever you want to do. So you have the metabolome and genome collected and preserved in a very small format within a very, very short period of time. So what do we do with this? And what we do, and what we suggest, is we can do 2 things. First of all, we 1 centimeter diameter discs. They're absolutely perfect, and this is on the bottom, to go inside 24 microclimate plate wells. So you can basically do any assays you want by just putting the disc with your biochemistry inside the microclimate plate wells. In this case, we just use the hematocidal assay, important for Africa. You put your worms, and if your disc is there, the compounds will be resolved, and you don't need to leave a loop. Okay, so it's very compatible with a variety of screens, or pharmacological screens you can do. Another thing, of course, you can do, which is on top, by eluting again, let's say 7% ethanol, because it's a rigid sample, you can do metabolomic profile. You can send it to you can put it in an envelope and send it to your colleagues. You can do it in the lab, you store them in the freezer. You can probably store whole plant biodiversity of Indonesia in a couple of freezers in this format. Just pull out the disc, do whatever assay you want to do with it, work with the metabolomic profile. So there's 2 things, then you can do 2 together. And that's really what we use that for. And just give you this a simple antimicrobial testing I'm showing you here. It's just MTT, which is a live staining. For example, if you want to test antimicrobial activity, we can use a 24 microcliter well plate with the disc at the bottom. You put your medium with whatever pathogen, bacteria, coli, Staph aureus, they will do. 10 hours, 24 hours later, you add your live staining. And if it turns blue, that means it's inert, that means no activity. If it's yellow, that means they have your antibacterial activity through live staining. And many other ways you can do it. This is how we teach working. Very effective, very fast, and you have your results. A high school student can do that. Certainly a university student can do that. We have trained when we work in Zambia and Botswana, we've trained the herbalists to do it because they always are very skeptical about science. And we brought the herbalists together with scientists and said, look, here's what you can use to validate the test. It was a very, very successful workshop. So there are many, many applications to this. So we actually now have a manual of about 23 tests, 23 different simple activity tests, functional tests, which can be performed in a 24 microcliter plate format using the RAMES discs again. Just drop the disc, you produce, and you can do antifungal and again, it's the most sophisticated tests you can do now, but that really gives scientists something to start working with. And because the speed of the assessment and the effort which is needed is important, it's very, very cost effective, it's very fast. And so this is so we have the metabolome preservation, and if you want, you have it preserved already in the format, just very compatible with any kind of functional assays. So why we are talking about Indonesia, why made Indonesia, and that's of course very important. We were lucky enough 4 years ago to get a large international program funded through the National Institutes of Health to provide research training in Indonesia in the area of botanicals and chronic diseases. And we have several partners. Pankasila University, University of Nacional were our additional partners. And then, of course, we met Annie, realized that so many activities also to work with brain and build. And so this has been a very successful program. We just actually had a symposium in UNAS last 2 days on botanicals and human health. We've trained about 25 master and PhD students in those 2 universities. 8 of them graduated. They got their diplomas from our program. International. They also considered Rutgers students. They have access to Rutgers resources, Rutgers library. Professor Navadi Sunaga is part of that, and also Singben Sator. We also have a program in Tajikistan, who is our partner. And the whole idea is to integrate the Western sort of science approach with traditional knowledge, which you know so much about in traditional plants. And last year, actually, we were able to get an additional supplement to add Ecuador, another country with enormous biodiversity. And actually, 2 of my former postdocs and graduate students now in the key research positions in the country. One was in the Ministry of Science and Technology of Ecuador. You can do the same thing in Ecuador. And it's only possible because we do so much of this online. Because we have synchronous and asynchronous courses, manuals, lines, and the medium of Zoom. So now we are no longer bound by the national boundaries. And next cycle, which we hope we'll get, looks like we are going to get the next cycle for another 5 years. We'll certainly welcome all other partners and all other collaborators to that. But this is dedicated more to study of botanicals and chronic diseases. Why am I here? Why we have been talking to Annie and many other colleagues? Because I think there's a possibility now, based on the resources we have, to establish what we call the Metabolome and Genome Innovation and Conservation Center, called MAGIC, in Indonesia. What will be the goal? The goal will be, obviously, we all know about deforestation, we all know about disappearance of natural resources, we also know about enormous land biodiversity and marine biodiversity of Indonesia, right? So can we at least start cataloging and preserving it? That's with the RAMES format. Basically, simple, right? Any student can do it, particularly focusing on endemic plants, indigenous plants, and rare plants in Indonesia. Just so we have a catalog, we have a voucher, and we can also very easily, if you want to study the metabolome functionality of these plants, just get the discs, use it from the assay, and just start creating libraries. And Indonesia will be the first country in the world which will actually catalog and create a library of the metabolome genome of its unique diversity, which will be solely owned, of course, by Indonesia. We will only use internationally with the discretion of Indonesian scientists. So, and we were fortunate. We got some more funding specifically for that from Asia Pacific Network, just to demonstrate the principle. It was the just a pilot scale program. We said we're going to try to collect 500 range samples, basically from different plants. And Asia Pacific work funded this work. And again, we've done a number of workshops, and several of my graduate students came to Indonesia to work with Britain, to work with UNAS. I think a lot of people now in Indonesia know very well how to do that. This is the second actually workshop we had in Kalimantan Salak National Park. It was wonderful. Antonio Kazda and Sarah Skubel there. Sarah is now a professor actually at CUNY. Antonio is still with me. Where we did actually in situ in the jungles. We tested the method, we collected samples. And now we succeeded in having 500 species of plants and then metabolome and genome preserved. This is the courtesy of Sudhir Navati. We organized the training and workshop of RAMES in Bali Botanical Garden. And this is where we're also going to symposium. Thank you so much. It was a wonderful symposium in Canada. So this is, I believe, both marine and university people who participated in that, and Antonio Kass from the lab was one of the trainers, and it was extremely successful. And I think what we did is validate the usefulness of RAMES technology to cataloging, preserving carbon material plants. This is just some pictures of actually creating this 500 examples in 3 different locations. Again, our advice is to work with, since we have such a great botanical garden system, right? It may be, and it's not destructible, only takes 1 leaf. The good thing about botanical gardens is that the plants are identified. Most of the plants are solely identified. So you know what you're studying with, and you already have thousands of species in botanical gardens. But of course, then you can extend to the wild. So this is just pictures of Isabel. The masses there. But I think by now, all the training has been done. So at this point, I think we all should talk and get together and see whether there is or really very little amount of money and effort to start collecting a couple of Indonesian plants for future research, for future discovery. And this is just been one, this of course, Amy there, who really arranged and organized this in a very, very wonderful partnership. And to me, yeah, that's a young kids, right? That's where you don't need to know much science to be able to do great GIBEX. And initially they all stand around, so again, there's people, and slowly they start seeing the results, the colors change, the activity appears, they come closer and closer, and suddenly they become fired up, they become excited about this, because you can instantly see the results if you combine it with yes, not very sophisticated, but still it's the first step on getting people interested in science and getting some very interesting antimicrobial activity and so forth. So what I want to just end with is to show you one example of what we actually done and published, again, using RAMES technology. And that was our work in the Kruger National Park in South Africa. We didn't want specifically to start collecting plants from South Africa, because again, they're international treaties, but we came up together with Dr. Lushenko about a very interesting project just to get to validate that technology. And one of the biggest issues in South African National Park is an invasive species. It's the plants that have been introduced from Europe and the United States 100 or 200 years ago, and which really found a very good home in South Africa, particularly in Kruger National Park, and they truly created environmental. They're very difficult to build. And this, so what we decided to say, well, those plants, they're not native to South Africa, so we can work, we can collect the samples. And at least 3 of those plants came from the United States, and they represent a major environmental hazard in South Africa. So we said, okay, can we just test the RAMES technology? Can we collect the metabolome of the native populations of those 3 selected plants in the US from different places? And then collect the metabolome again using RAMES technology in the field from the trunk of the car. Can we collect that in the population which invaded Kruger National Park, and then do this following analysis? Just compare whether we can tell that whether plants 200 years living half the world away under very different stresses and different diseases, disease pressures, did the plants change their metabolic profile? Do we just non-targeted metabolic approaches? Hello, and Prof is a key botanist in Kruger National Park. He hosted us, and we selected 3 different plant species which are invasive from US, Propolino, the radar, Daturus ammonium, which this is all taken in South Africa, and Xanthium strumar, which is 3 plants, because okay, those are not native plants of South Africa. We can collect, we can use RAMES, we can collect this. And so we spent 2 years collecting samples, again, on RAMES discs from the South African populations and from the populations in the United States, which have been separated artificially for at least 100 years to see what they adapted. And this is just Sarah and Lowland and then Ranger doing the extraction. We find, we just drive around, find plants, we quickly extract them, put them on the discs, and that's it, we're done, right? So then we can analyze them later on. This is how they look like. This is For the typical, the collection is the plastic bags, each sample is labeled, with GPS coordinates taken. And I don't want to bore you with the data because it's been published in PLOS One. Again, and the main ways you can analyze the non-targeted metabolomic data, but the results are very, very dramatic. Okay, if you really look at the quantitative and qualitative, the population in South Africa were very different from the population of the same species growing in the native habitats in the United States. And for example, this is a partially square regression analysis. You can see how South Africa, which is blue, and US populations, just based on the metabolomic profile, how dramatically different how those plants adapt. And when you look and identify the compounds, you see that there are many compounds which are present in South African populations, but not present in the US population. So plants are actively adapting, they're actively learning. And I think if we were not to use the RAMES type approaches, that would be a very, very different, very much more complicated way to analyze it. Just extracting grams of materials carried in plastic bottles, water evaporators, and all that stuff would be very, very difficult. So yeah, this is also published a little bit online. We actually published the papers on that. What I want to say is how grateful I am for the help which Green has provided in this particular project, and then specifically provided UNA's funding, so of course, that's the system health and APM. And really would love to see whether we can build on this 500 samples which already been collected and start this economic library plans to use the RAMES format using this class 500 disks. So what they can be used for future research, discovery of the compound. Yeah, this is the students, our students, and UNA students, which have been so instrumental in so many things which we do. And of course, this is Professor Tushko, his field assistant in Penching National Park, where we spent wonderful 15 days. And this guy just came over and sat next to Dr. Tushko. I think he just had some fondness for him. And so Slavic has been.
It's not Photoshop.
No.
And I'm with the left.
Slavic was on the left. So, and again, it's just like I love nature, I love nature photography. So I'm very blessed to work with the media. So that's my part, and this is what we are. And I think it's good that we're having Paula here, who's also an expert in analysis and research, and maybe also join this effort to start creating more libraries for the future generations, because some of those masters will be here. Thanks.
Thank you, Professor Raskin. Very interesting, but please be patient because we have another presentation first from Professor Tushko of Slavic. Then after that, I will present maybe like 2 or 3 slides about RAMES. So this is nasi rames. Do you have in Indonesia? We have a kind of question or dish. Nasi, you know, right? Nasi is rice. Rames. Rames is exactly what you read. Rames is, you know, compilation of many dish. Fish, meat, vegetable, and of course sambal with chili. And this morning we discussed about chili pepper. Yeah, so we call it rames. So this one grapes. Okay, so thank you again, Raskin. So here, please, if you don't mind, Mr. Slavic, and then later on we will have discussion, presentation. Can you see the slides? The online one?
Belum di share. Zoom.
Can you see the slides? So the back is yours. So how you can.
Yeah.
Thank you very much. Of course, I want to thank the organizers for inviting me. It's a pleasure. It's not the first time I'm reading, but every time I'm coming to this wonderful facilities and this wonderful campus, I'm super impressed. So it's an honor to have opportunity to talk a little bit. And what I would like to do today, I mostly would like to introduce my university, which is much younger than the university, and give an example of how we actually put together the international collaboration, but mostly to introduce my university group. Okay. It's a young university, just only was organized in 1847 due to vision of Mr. Harris. He was a reader, and what he wanted to do, he wanted to bring to New York City the university which will actually educate all people, spend the day in it was designed to educate the population of the New York City, and especially targeting the population with very limited income. The first campus was in 1947, and you can see this on the right. This is the City University of City University of New York, and City College campus, which is okay, good. That's good. Oh. Yeah, I will not get going. So Mr. Harrison was a very interesting fellow. In addition to organizing the university in New York, City University of New York, he for a long time was an ambassador of the United States in Japan. And he still is very much honored as a collaborator, bringing those two countries, United States of America and Japan, together. What you see in the right bottom bar is the City College campus. It's one of the major campuses of New York City University. A little bit of statistics. City University of New York has 25 different campuses all over the 5 boroughs of New York City. On the map of New York City, you can see location of the campuses. Many of them are in Manhattan, the central part of New York City, but they also are in different boroughs, in Bronx, in Queens, and Staten Island, as a separate campuses of the university. It's one of the largest universities for university in the world, with 500,000 students. 500,000 students. Actually, there was a sort of friendly argument in Boston and New York majors quite a bit ago in terms of which city is better than education, and the Boston, known as a university city, the major of Boston said, well, no, we are the famous Boston University and Boston educational city. For that, the major of New York said, well, that may be, but you know, I have more students than the population of Boston. So indeed, it's only one of the universities in New York City, but 500,000 students, it's an impressive number. It's an impressive number. We have
1,750 different degrees program. University brings annually about $500 billion, that's 500 million US dollars in research funding. And we're proud to say that the City University of New York is a non-matter for 13 Nobel Prize laureates and 26 MacArthur Genius Grants. So it's a well-known university with a lot of efforts. About 20 years ago, the question was, okay, we are bringing so much money in the research funding, what are we going to do with it? How is best to organize the research in the city university? And of course, traditionally, it's a variety of different laboratories which their own research. And that time, the visionary provost of the university, actually a vice chancellor of the university, Dr. Small, she argued that it's better to put the money in something that can be concentrated and can bring together the resources to build a state-of-the-art research center. And that was done. The One Science Research Center of the City University of New York opened on the campus of City College, I think it more than 10 years ago. And at that time, it was, that's the building actually. At that time, the university started to put in a lot of money in building the facilities, building the state-of-the-art instrumentation, and the best possible teams to run this instrumentation, with the mission to serve the City University of New York researchers and outside the world also. The university decided to concentrate on 5 different programs. It's environmental science, 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 photonics, and structural biology. Those 5 major directions of research are still part of the advanced science research. Environmental science has wonderful opportunities, all the labs for all over the world. And I would like to highlight only the most recent achievements. It's not necessarily the best, but it's one of the most recent achievements that each discipline is known of this year. So the scientists of the environmental science group, along with other people from the university, put together a wonderful program analyzing the water quality and water resources worldwide. And those water resources worldwide are basically in very good shape. And the research provided opportunity to see where the problems are and how to address the problems. Non-science, actually the field director of the center, and it's actually targeting this basic merger of biological and physical science on the nano level. One of the latest discoveries of this group is that in addition to structural proteins, there are proteins in the cell which are much more flexible. These all flexible proteins are creating like a small factories where the majority of the chemical reactions are going. Those liquid droplets play an important role in the cell, but they also have this opportunity to be a small protein, flexible, with very fast changing properties, to be the basis of nanotechnology we use in medical science. Neuroscience. The whole division is what you almost and one of the latest advantage recently just published is finding that allegedly proteogenous cells, that the cells which are creating other cells in the neural system, and which are responsible for providing the ganglion power for the neural cells. And then the basis of many diseases, the destruction of this coverage, and then the opportunity of bringing this cell together and restoring the coverage. And that would be used in a lot of medical conditions, including the Alzheimer. Photonics, well, anyone like they showed the photonic there, they're dealing with the waves, light waves, sound waves, and that's one of the waves, the highest money maker of the center. Reaping a lot of grants, especially in this case, applies what I'd like to bring your attention to is that the recent advantage in using the imaging processing technology to make the driverless cars and the image processing much simpler, saving a lot of energy and bandwidth for this technology. And finally, the structural biology. Structural biology is, in the main, when we're going to proteins, most of proteins, it's a very close collaboration with different university groups. And they discovered a new method of computation and analysis, X-ray crystallography, which found a variety of sites. So instead of old-fashioned techniques that was used to characterize proteins for the last 10 years, we can actually advance it. And on the right, you see the graph how this advanced new crystallography provide opportunity by 4 sites here. For the source, still 5 directions that is not signed at the center are still very active and producing a lot of wonderful results. However, of course, we're interested in something which is close to our programs and the facility that provide advanced science research center. The center actually obligated, most of the research within the center, but obligated to help all these 25 campuses of the City University of New York and outside to do research by using the specific, very high care instrumentation. And facilities are listed there quite a bit. Facilities, we are something that can help us to analyze the particular chemicals and metabolic analysis also. Here's just a few examples. We have the facilities that's in the electromagnetic resonance. 5 years ago, it was the best in the world. Now they get even better, but still very good facilities that can grow. We have imaging facilities, which is a combination of a variety of microscopes, state-of-the-art microscopes, and something which is becoming even more important than microscopes, the hardware itself is the software and analytical tools. And that allows you to do a variety of microscopy from actually atomic force microscopes to live cell microscopes and animal live microscopes. Spectrometer facilities, that's what we are interested in. State of the art. Initially was designed and put together to analyze the proteins and variety of protein structures. I have to take some credit on bringing in asking them to do the plant material. Also, for the last 7 years, this facility is very actually along the analysis of the cell. Well, that's what we have. And that's what would be used. And then the facilities and not just on the facility, but the expertise of the center could be brought to the board national operation. You showed us level where GIBEX is active. And that would like to use it only to demonstrate one of the examples that we use this opportunity to watch collaboration on the different topic. Central Asia, and there's a Central Asia, Central Asia, and here is the Kyrgyzstan. Kyrgyzstan is a small country, and 93% of this country is high altitude mountains. So all the 7 million population mostly work and live in all 7% of the country. And being an high altitude country, being in the center of this mountain area of the Central Asia, and being on the way of the Silk Road, which is also the Kyrgyzstan became one of the biological centers of the Central Asia, where a lot of plant material was moved all around together and so on. One of the plants that we paid attention to is the Hirula, with about 80 different species worldwide, concentrating mostly in the Central Asia and Mediterranean, in the North Africa, and Middle East. Hirula one was used from, it's actually mentioned, I think, in the Bible also, but it's used forever, mostly as a spice. However, it's also used in medicinal purposes. In the Kyrgyzstan, there's 27 species of Hirula plants, out of which 6 species which are listed here are endemic. So we discussed with the scientists what we can do about this. And the idea was some of the species of Hirula are very well known. Concentrate from the endemic species which nobody actually analyzed their phytochemistry. Species that are usually used in Tajikistan for medicinal purposes, but not very much known about those species. So we took those species and did analysis of which during the whole time the interest was in antiviral properties. And we found that one of the endemic plants, the Hirula violets, had very impressive antiviral properties. And that's published material. So now only I'm getting published material, but just to give you a idea where we are moving. And it's just the beginning of the analysis, so it's not the final termination. So we took the Hirula violets and plant, collected them in the fields, and the City University of New York, the facilities to the on-target methodology. And that's again, that's a work in progress again. So what we were able to show, we were able to get up to 5,000 peaks, which after the very careful analysis was in control, was reduced to about 2,500 species. In the samples that we brought from the previous time, the least something like 2,500 different chemicals. And when we start to look at those, we found that many of them are not reported for fruit species. And in this table, you can see that we're analyzing, still analyzing the different national chemistry pathways. And we are finding that a lot of this material that we are getting from this species are new for Hirula, and sometimes maybe the plants. Subclass distribution. You look at the subclass distribution, there are a couple of surprises here that we were actually very, very interested in. When we look at the total chemistry one of the things that strike us, and we're still trying to figure out whether it's true or not, and it seems it's true, enormous amount of alkaloids in the plant. And the distribution of these alkaloids is a little bit different in 4 different samples that we're analyzing. We're analyzing roots versus seeds, and we're analyzing 2 different extraction methods. One is the standard ethanol extraction, another just juice uptake from the raw material. It seems that extraction method affects what results you are getting, which is no surprise, but we're trying to get to the bottom of it and to figure out what actually it is. So this work that I showed today is a result of collaboration of Tajikistan, which has this unique genetic plan, Kazakhstan, where the majority of the antiviral activity was tested, and United States, where we put Prof. Of the 3 countries, experts from 3 countries, which are the best and they are all people, put together to do this project. And that's how we are working collaboration with GIBEX. Thank you.
Oh, thank you very much. Yeah, it's very insightful and I think giving us inspiration, especially for young generation. Okay, we only have 15 minutes left on this is Friday, but we can have a little bit more. So yeah, I've visited the CUNY, and CUNY is just like breathing, but only one breathing. In here, we have to run around in several buildings, but there, everything, there they have currently 2 sets. And all, yeah, like the one you mentioned is Rima. I met them, I met him, and all other expertise. The photonic also very good facilities because someone from Physics Research Center met me and then want to do work together on photonic. They also designed photonic, but very simple one. So yeah, I will discuss later on. I just let me thank you again. Just want to share 2 or 3 slides that we've done for RAMES. Friends. Same path. Okay, since we got the machine, the one that Professor Raskin showed to us, so after the Valley Botanic Garden training course, then we got all the machine and equipment, consumables and reagents, and we did several tests using our resources here. And up to now, actually, they came here, they brought us the chemicals and the reagents because we run out. So still, this is kind of income value we can value, but $1, but they brought thank you. So this one we did the extract from the Botanic Garden, Botanic Garden, and here surrounding the Sukarno Science Center and Technology Center, and then Sungai Wayne Protected Area Forest in East Kalimantan and Gunung Ciremai. Gunung is Mount Ciremai National Park border area. So this one, yeah, it's good that we are in the same chemistry, Prof. Raskin. That I showed this one also, one of also your slides, same one I put here and then over here. So we collected in the Botanic Garden, and also some of us also attended the branch of technology. And then here, also in this area, we did that, so we involved students under our research groups from Defense University and other university in North Sumatra and in Jakarta, Raffles University, and we did that, and you can see the different color and dark or white or something. And then we asked one of our students, the Defense University, when they did the 1 or 2 months in the field, they have to serve in Indonesian university student. They have to serve to live in community for 1 month or 2 months. We call it KKN. So this one, because it's a Defense University, so Cinta has to dress also with like army. And then this one I connected with one lecturer and researcher from Universitas Mula Warman. It is Kalimantan. Why is Kalimantan? Because Kalimantan is the area of the new capital. So then we tried all except like this. So she brought it under the rucksack because they only allowed to bring 2 rucksacks. And then this 9 species, and then this the list of species and the pests. And then on the activities conducted, also Cinta tried to use a different kind of organs, root, stem, bark, and leaves. And then about the functions, and then we also tested from Gunung or Mount National Park. So comparing the flower buds and others. The lab, so that's it turns out that the one from Majalengka is better than the content than the one from Kuningan, because there are 2 districts in the area. So this kind of thing. So this again, yeah, why is killing you? This is just like weed, but it's got a lot of content for health related hormones. So this I recommend to people because we need to have some equations, and then keep it also kind of weed, but also a lot of metabolism and compounds. So in fact, this one, so the idea of RAMES is to low cost and very efficient and very quick to go to further research. Yeah, so back to the lab and so on, and you don't have to bring the samples outside the national park because it's a conservation area. We need to have certain permits from the Ministry of Forestry. And then Professor Raskin already mentioned about the 500 RAMES collections. It's all kept in being kept in Indonesia. So it's not going outside of Indonesia. And then we analyze for further analysis for metabolomic or genomic later on. So this one is shortcut, and then you don't have to do very extensive analysis with HPLC or with LCMS. Try this one first to do first screening. So yeah, I think that's I want to show you, and then before question and answer, or very limited time, I would like to give the opportunity to Dr. Brown, maybe 5 minutes or so. Yeah, absolutely. Yeah. Just put your. You want to sit here? Yeah, because so that everybody can see your face. So I will do that. They can.
So thinking maybe it's